So I'm just waiting a little bit to get everyone seated, and we have another day of great presentations. And the first two presentations this morning are going to be associated with cyanobacteria. I think it was almost 10 years ago when Russ Kantz from the State Board brought this to our attention. It was on the Klamath. The two speakers we're going to have starting us off this morning, one of them, Dave Crane, you already know, but the other one, where's my pages there? Kim Ward. She's been working on this quite intensely just recently, since 2006, working on cyanobacteria problems in general for the State Water Resources Control Board. But was working on an interesting project in Watsonville, which I know the area, and she has a grant there to talk about the treatment of cyanobacteria in a wastewater treatment plant, how to get rid of it and not the other beneficial bacteria and such. So we're going to get started off with Kim Ward, and then she's going to turn it over to Dave Crane. Kim?
Good morning, everyone. Let me know if you cannot hear me in the back. I sometimes am justly accused of having a very soft voice. Now, some of these slides, I'm going to admit, are in microprint. Don't even try to read them. These slides will be available online, I believe, and archived if you want to read all the gory details. So I'm going to run through some of these and try not to dwell too much on the details, but just give you a general glimpse of what the literature seems to be saying these days about aquatic toxicity in reference to cyanotoxins. Overall, people who've looked at this issue intensely, like Hans Perl and his colleagues at the University of North Carolina, have noticed that these so-called cyanohab blooms have been increasing globally. Reports are coming in from literally every continent. They seem to be on the rise. The toxicity associated with some of these blooms is also notable, and research on the general issue is rather intense. It's a burgeoning area of active research, and more toxins continue to be identified. And when I say toxins, I mean substances that may be toxic to mammals, vertebrates, invertebrates, even some green algae and higher plants, including aquatic plants. And that's why the general ecosystem question has arisen about what are these toxins doing in the environment and how are they disturbing what might otherwise seem to be a peaceful bloom. At present, we have no national surveillance program for these cyanohabs. There's a passive reporting system that the Centers for Disease Control in Atlanta has been putting together with no money. And it's really focused more on what you might call the human health or mammalian toxicity issues and or recreational water body postings and closings. And no doubt you've heard of what's been going on in the Klamath perennially with the blooms up there and the posting issues. And if you have, that's just a microcosm of what's happening in some other water bodies throughout the world. But first, I think I need to clarify what cyanobacteria, sometimes known as blue-green algae, are. It's not only not technically correct to call them blue-green algae, it's also kind of misleading. Because they have a number of different photosynthetic pigments, they can appear in many different colors, from green through blue-green and magenta and orange and yellow and red and just about any color you can think of. Sometimes they appear almost black. They're also not eukaryotic algae, and though they tend to coexist in mixed blooms and are a normal part of the aquatic environment, they are not biologically to be confused with bacteria. They have a very different way of pursuing their particular natural history. They tend to be very tolerant of high pH, high turbidity, elevated water temperatures. Some are true extremophiles, meaning they can survive in geothermal hot springs. Some can survive frozen literally under icy lakes in Antarctica, and some grow sometimes in the rocky crevices of rocks. Sometimes they're endosymbionts. They can actually be found inside of other organisms, photosynthesizing away, it would appear. Some are also symbionts. Lichens would be a good example. Overall, they perform a lot of very important eco-services for our environment here on this planet. They are very important in the biogeochemical cycling of nitrogen. They pump a lot of oxygen into the atmosphere, and it's estimated that 50% of the photosynthesis going on in the oceans is attributable to them. They may have, at one point in the remote past, actually been the inventor, if you will, of the chloroplast that we find in all the higher plants. The chloroplast is where the action takes place. This is where photosynthesis occurs. It may be that at some point in the remote past, they contributed genes to what we find now in the modern chloroplast. At least that's the way the current research seems to be indicating. So for 3.5 billion years, we've seen innumerable really, episodes of horizontal gene transfer among related taxa and constant mutations, giving rise to a number of different strains. Now this is just a comparison between a chloroplast, a modern one, and a cyanobacterium that you may want to review at your leisure if you're interested. It shows you that the various structural elements are nearly identical when you compare the two. Here's an idea of what it looks like when you encounter these blooms. You may note that there are vast color differences. The one on the right is a marine bloom. It's trichodesmium, and it's kind of a dark red in this case. It'll vary quite a lot. Sometimes it'll look like straw, and sometimes it's called sea straw. It also produces toxins. The one on the left is from a freshwater lake in China, as it happens, and there's an aging bloom, and you can see the various colors, kind of an artistic pattern created by the bloom as it dies off and the pigments start to fade and bleach out. Here's another example of mixed blooms. This one on the left is in a geothermal pond in Yellowstone, and the cyanobacteria happen to be the yellow, orange, and red part around the outer perimeter. A halophilic extremophile, which is also tolerant of very hot conditions, is the blue stuff growing in the middle. And on the right is an Antarctic lake where a diver is showing you a mixed bloom in a mat. It's forming literally a sheet across the bottom of an Antarctic lake. Overall, the cyanobacteria, although they are not aerobic organisms, they're anaerobic, they can still outcompete in some circumstances. For example, they're able to outcompete green algae for things like nitrogen and phosphorus. And as mentioned, they are indifferent to high pH and low oxygen because they're anaerobic. And for them, ammonia is not a toxicant, it's a source of nitrogen. They do photosynthesize more efficiently in turbid waters, and they engage in what appears to be a strategy of chemical warfare with other phytoplankton in the water column, potentially with zooplankton as well. When nitrogen runs short, some groups can actually fix nitrogen from the atmosphere. Now, this is a very crowded slide, but I'm going to skip through a little bit of this rather quickly. You can read the details if you wish later. As mentioned earlier, researchers are finding that these blooms are getting worse, they're more extensive, they last longer, and they cover larger areas. And this is a phenomenon noted worldwide. Anthropogenic inputs into these situations, including, if you will, climate change, to the extent that that's anthropogenic as well, may be contributing to these blooms. It certainly seems to fit what we know about their general ecology. One result may be what we're seeing in a recent survey done of thousands of lakes here in North America. In 2007, the National Lake Assessment Survey was conducted. 42% of the samples exceeded, for one particular type of cyanotoxin, which is called microcystin, the 10 part per billion level, which is kind of significant when the World Health Organization is saying for drinking water it probably shouldn't exceed 1 part per billion. 30% of the lakes total had microcystins. Other cyanotoxins were found as well, though not in quite the same abundance. Some of these included saxitoxins. Some of this group may be usually thought of as marine toxins, but saxitoxins apparently are also found in fresh water due to cyanobacteria. Another cylindrospermopsin is previously described as being tropical, is now found throughout North America. Anatoxin A is a noted neurotoxin. Nodularin was thought not to be present in North America. Now we know it is. And here's a picture of what some of the blooms look like locally and not so locally. On the left is a recent picture taken in Clear Lake Oaks, Keys, And on the right is an irrigation canal in the UK. I thought you might find this helpful in understanding how, in some ways, anthropogenic activity modifying the hydrology of an area can create kind of a culture flask for these blooms. Add some extra nutrients, and you can get situations like this. Here's another crowded slide, and I'll run through this briefly. The pharmacological potential of these toxins walks hand in hand with their toxicity potential. Indeed, some of these toxins are under active research to explore them as potential antibiotics, some as chemotherapeutic agents, and so forth. The toxic effects noted so far can be acute, they can be subchronic, they can be chronic. Increasingly, as people investigate these very different groups chemically, they're finding that some can actually bioaccumulate, and that, of course, poses some ecosystem questions about where they wind up in the environment. Microsystems are notable in that regard, but so are nodularins, SWRCB, and we got about 17 members of the so-called paralytic shellfish poisoning or saxitoxin group, too. I'm not going to dwell on human health and mammalian exposure. That literature is pretty well covered. You may all be familiar with it to some degree. I'm going to focus a little bit more on organisms you may not have heard about yet with reference to aquatic toxicity. But briefly, some of the exposure pathways for humans and mammals include aerosols. That may be one of the most dangerous ones. There's food and water, there's dermal, and then there's intravenous exposures. For example, there was a big problem in a dialysis clinic in the early 1990s in Brazil, and dozens of people died as a direct consequence, it would appear, of having these toxins placed directly in their bloodstreams. For us, they're colorless, odorless, and tasteless. I don't know if other organisms are picking up on them directly through these mechanisms. Some are cyclic proteins, and the reason that's important is because they're very stable. And for instance, the microcystin group, all 90 plus of them, are resistant to heating, and they're also resistant to freezing. They can be recalcitrant to conventional water treatment technologies. And because they are bacteria, you can see resistance in this group developing rather rapidly. So if you treat a lake with copper sulfate or hydrogen peroxide, it's been noted that resistance tends to occur even to these very strong agents that you might not think organisms could respond well to. As mentioned also, they can transfer genes around, including toxin genes. And when you do run toxicity studies, the classic test is the whole cell extract in a mouse bioassay. You usually get more toxicity than you can account for based on the toxins that you can identify within that group. There are about 3 major modes of action in animals and humans: neurotoxins, hepatotoxins, and dermatotoxins. I will say briefly, however, that it's starting to look like some of the hepatotoxins could be neurotoxins as well, and even cardiotoxins. Microcysteines are suspicious. They're also very common, so I would say keep an eye on that one. Now, this was completely illegible. I throw it in here for reference, but it came from a paper by Castle and Rogers, and it's one of the best breakdowns of both the chemical identities of the known toxins and the major taxa which are known to produce them. That list grows with every passing day in both cases, both the list of toxins and the suspected taxonomic groups involved. This one comes from Keith Lofton. He's a chemist with U.S. Geological Survey. He gave a presentation last year, and I liberated a copy of one of his slides. At the very bottom, you can see, and it may be hard to see, there are acute LD50s, lethal dose 50s, milligram per kilogram body weight. And at the very bottom, you see ricin, and then along to the left, you see saxitoxin. You may note they're not very far apart. Ricin is included for reference. What Keith was trying to do here is show you that these are first-class chemical warfare agents at some concentrations. And then on top you have SWRCB. It looks relatively innocuous. It's only as bad, more or less, as rattlesnake venom. I need to say a little bit more about microcysteines briefly. It's not only a hepatotoxin, it can be a tumor promoter in reference to liver cancer, we think. It can bioaccumulate, as mentioned. It's stable. And it's the best studied of them all. So there's a tendency to focus on what you already know, and I think in the literature we're seeing a lot of that. A lot of people are looking at microsystems, and now we're up to 90 different structural variants, some of which seem to have rather different properties from one another. So when you talk about microsystems, it's hard to make generalizations. We certainly haven't tested them all. Only a handful have actually been subjected to a lot of toxicological work. When we step it out a little bit away from mammals and humans, we start seeing other potential effects. And for example, in aquatic environments, you might start seeing effects on fish and birds and invertebrates. You might see problems in aquaculture. You might have effects on domesticated animals and possibly irrigated agriculture. This is another illegible slide. This comes from basically a lot of intensive work that OEHA, that's the Office of Environmental Health Hazard Assessment toxicologists, did on contract with my agency, the State Water Resources Control Board. And I had the pleasure of working with these wonderful toxicologists and poring over the literature up to 2009. And we only, unfortunately, had enough funds to look at some of the toxins, so we tried to prioritize the ones that were both thought to be the most dangerous and also the ones with the most extensive literature. So we wound up looking at a handful of microcystin structural variants. We looked at anatoxin A and cylindrospermopsin. Now, it's all but illegible, but I will point out in passing that we looked at different endpoints other than human drinking water. In general, Department of Public Health looks at the environmental risk associated with drinking water, so we looked at other potential endpoints where we thought there was genuine cause for environmental concern. So for recreational uses, we have action levels, human fish consumption, subchronic water intake for dogs, which, by the way, turned out to be one of the more sensitive thresholds recommended. Subchronic crust and mat intake, yes, dogs and cattle seem to have a preference for actually consuming these organisms, whether they're toxin containing or not. And the threshold for dogs for some of these toxins is among the lowest on this chart. Again, this is illegible, my apologies, but we do have it on our website and it will be included in these slides. So when we look at cyanotoxins in aquatic food webs, we note that even carp and other fish that are known to be phytoplankton grazers can, at some concentrations anyway, be poisoned by various cyanotoxins. We also see that zooplankton, oysters, mussels, and other shellfish and miscellaneous invertebrates can bioaccumulate some of these cyanotoxins and toxins, and in some cases may also exhibit signs of toxicity. This is a very interesting area of research. Not a lot is known yet, but the preliminary indications are that this may turn out to be fairly significant. I took a look at what's been published within the last 3 years to see what I could glean about aquatic toxicity non-mammalian. And as Corbell and other authors who did a review paper this year were saying, that there's a lot we don't know, but there are some indications that something's going on. Microcystins, for example, can impair plant physiology and metabolism. Cylindrospermopsin can inhibit plant pollen germination. Anatoxin A can be a stressor for some submerged macrophytes. Microcystin can also reduce the activity of photosystem 2 in green algae. This may be part of their chemical arsenal for competing. Microcystin also can reduce apple shoot growth in vitro at 0.03 micrograms per milliliter, and that may be significant for agriculture. When you look at Daphnia, you find many different species, of course. With one tropical species, recently some researchers have identified that microcystis could actually reduce the growth and survival. However, it was not related to the fact that microcystins could be present. So there seems to be some toxicity with some strains that has nothing to do with microcystins as far as some Daphnia are concerned. If you take an extract from Planktothrix blooms, you find that they may act as an endocrine disruptor, at least on the Medaka, which is a fish frequently used in toxicological testing. Anatoxin A, usually known as a neurotransmitter disruptor, at least invertebrates, also seems to work its magic on rainbow trout. Motor impairment seems to last. You get reduced acetylcholinesterase activity in Canadian freshwater amphipods that were collected from Lyngbia mats growing in the St. Lawrence River. So that gives you a brief overview that there's some areas out there with some question marks. When we look at the 303D listings in California, we find that there are about three major reasons why we have listings that are associated with bloom problems. One of the reasons we'll see these listings is because of alterations of our natural watershed hydrology. We have reservoirs, altered flow regimes, and water diversions, for example. We have multiple sources of anthropogenic and sometimes natural nutrients. We have multiple anthropogenic sources of phosphates adhering to sediments. And as a result, we're starting to see some areas where these blooms have been documented to be causing problems. These include the Klamath watershed, Eel River, Big Lagoon, Lake Isabella, that's in Southern California, Salton Sea, Clear Lake, the Sacramento-San Joaquin Delta and Estuary. Pinto Lake is in Santa Cruz County, not far from the ocean. It drains ultimately to the Pajaro River. We have sea otter poisonings along the entire Monterey Bay shoreline and south of there that are confirmed to be due to microcystin. And we have several Southern California locations, including reservoirs and lakes, where cyanotoxins have been found. This is just a map from a recent paper that Melissa Miller, Dave Crane, and lots of other people have worked diligently on, and it culminated in a publication in 2010. And this map shows the work that people like Rafe Cadella and his graduate students did trying to identify where were the microcystins coming from in the Monterey Bay watershed after finding these sea otter poisoning cases distributed up and down this area. And it did look as though it was emanating from freshwater sources. Subsequent work confirmed that, indeed, microcystin production is year-round in this set of watersheds. Although it waxes and wanes in concentrations, it's never not producing, it would appear, and there seem to be multiple sources. It also maps, interestingly enough, with the locations of where some of the dead sea otters that were poisoned due to microcystins were recovered. As mentioned, we have numerous areas in California that have blooms, and that has resulted in some listings under our 303D program. We see effects on what we call beneficial uses, and we see a variety of impairments of these so-called beneficial uses. We see impairments to fishing, invertebrate harvesting, cultural resources, recreational resources, drinking water, and wildlife. And that concludes my presentation, and I hope I haven't run too far over time.